Hi, this is Mr. Matthews, and this is the podcast on DNA and your notes on DNA. So, before we get into the history and the structure, it's important to talk about why we even study DNA. We've spent a considerable amount of time in class going over these, and these are all the lists of reasons why. The Human Genome Project is a recent study that was completed just a little bit after 2000, so sometime maybe even in your lifetime. And what this was scientists mapped out every single gene in the human body, and this is going to have profound implications for medical reasons and technology speaking when it comes to DNA. DNA identification is another one that we tend to look at, not just in crime scene investigations, figuring out who can be linked to the scene of crime, but for paternity cases, whether a child belongs to a certain mother or father. Cloning, we've spent quite a bit of time looking at cloning sheep, cloning dogs, cloning cats, and what's even stopping us from cloning humans. And I guess one of the many few video clips we've watched on cloning begs the question, just because we can doesn't mean we really should. And that's an important question to kind of keep asking yourself when we deal with a lot of what's going on with the DNA technology. Gene therapy for medical purposes, being able to create genes um, to replace some of the, if you have a certain genetic disease, to create genes or to grow genes in the lab and to replace the genes that are wrong in your body with these genes that are grown in the lab. Genetic engineering is a huge topic, manipulating genes for human purposes, whether it's for food or vaccines or drugs or any kind of other reagents. So there's a whole bunch of really interesting reasons why it's important that we just study DNA. So if we look at the history behind DNA and some of the scientists involved in researching it, we start with Miescher in 1869, extracted a white substance from the nucleus, and he called it a nucleic acid. Chargaff came along later in 1949 and found that this nucleic acid had some nitrogenous bases, and that the base A pairs with T and the base C pairs with G, adenine and thymine, cytosine, and guanine. This is known as Chargaff. So we'll get into that when we really get into the structure of DNA. But he's definitely important to note because he has this rule that these bases pair up in this nucleic acid. And then we go all the way to 1952, the Hershey and Chase, and they worked with mice and they proved that DNA is indeed the genetic material. They worked with viruses and bacteria in mice, and that's what they came up with. And they were very instrumental in Rosalind Franklin and Watson and Crick's a race, and it really was a race to come up with the model of this DNA. Franklin and Wilkins worked together in King's College, and they did an X-ray diffraction of and suggested that DNA was a coiled spring shape. And in fact, um, uh, Rosalind Franklin was a woman, of course, at the time that went through an enormous amount discrimination, just being a woman and being in the lab, and the names that people used to call her, and some of the hoops that she had to jump through is truly amazing. And if it weren't for her actually dying of cancer, she would be much more famous than Watson and Crick, who we found out later that they really kind of took Rosalind Franklin's ideas and ran with it and came up, used her information and determined that the 3D shape of the molecule was in fact a double helix, and they came up with their own. They did this, they really took her information without Franklin knowing, which generally we call that stealing. And in science, it is customary to share your information, and a lot of scientific discoveries have been as the result of people sharing information. But this information wasn't shared. Watson and Crick basically stole her information, or they were given her information from someone else, and they used that to their benefit. And so Watson and Crick became famous. Rosalind Franklin just became another scientist studying DNA, and the rest is history. She ended up dying of cancer, and so she's been unable to really defend herself throughout the course of the years. If we look at these bases that make up DNA, the adenine, the thymine, the cytosine, and the guanine, they generally occur in the same amounts in all organisms. Since A pairs with T, you're going to have the same number of A's as T's. And this is just an example of different organisms and how many bases that they have in a strand of DNA. Again, you can see the numbers pretty much match up. The A's and the T's are about the same number all the way down. And then the C's and the G's over here, because they pair together, they basically have relatively the same amounts in each one of them. Um, so when we look at what is DNA, DNA is deoxyribonucleic acid. And it's the blueprint. It is the code for all proteins in our body. And it's the proteins that really determine what we look like. And that's the same for all organisms. And that includes like enzymes and hormones and structural proteins. But it is the blueprint or the code that really makes us who we are. And that's what all these, flipping back here, these A's, T's, C's, and G's, that is the code. That codes for proteins. So if we look at what it looks like, we did this in the lab already, and this is a picture of what DNA really looks like in a macroscopic view, meaning that we can view it with our eyes. And here's more of a computer-generated model, and you can see it's a double helix. It's a twisted ladder, a double helix. We see the bases in the middle, and the outside we have the sugars and the phosphates. And we'll get to more of that structure sort of later on in the presentation. If you look at the size, an average cell nucleus is about 6 micrometers in diameter, and the length of a DNA in the human genome is about 1.8 meters per cell. So if you can think of that in one cell there's 1.8 meters, that's quite a long strand of DNA. Multiply that by the billions of cells that make up your body. We're talking about one human being being able to stretch out one human's DNA like all the way to the moon and back, and it's probably even further than that. But it's a lot of DNA that we have inside our body, and that's the whole point of really talking about these numbers. So if we look at the structure of the cell, and again, if I'm going a little too fast, that's okay. You can pause it and you can write the information down, or you can stop it and come back later. But so again, if we look at the structure of this DNA molecule, structure of DNA is composed of units called nucleotides. There are 3 parts of a nucleotide. Let's flip to my slide. There are the bases, there are the sugars, the deoxyribose is the sugar, and there's a phosphate group. And I'm going to step away from this presentation and just go to another one real quick because I feel like I can better visually show you what this molecule is really all about. So if we go to this one, all right, and this is again the structure of the molecule. We have a sugar, which in this case is deoxyribose. We have a phosphate molecule, and then we have one of the bases, A's, C's, G's, or T's, one of those bases, and you put that is a nucleotide, one nucleotide. You start stacking up these nucleotides, and you get your molecule of DNA. And then that gets twisted up into our into chromosomes, and this is what is found in our nucleus. So this is a really good picture of what a nucleotide is and what the DNA molecule really looks like. So we'll come back to this in just a few minutes, but if we want to flip back to our notes here. Here's the structure. And then here are the bases. There are 4 nitrogen bases, adenine, thymine, cytosine, and guanine. All right, and they're also known as complementary to each other because A pairs with T and C pairs with G. And if we flip back to, again, that other presentation, these are the 4 bases, and you can see it's just like a puzzle piece. So A and T pair together, right? They bond together. The C and the G, they bond together. So that's what we call complementary bases. So moving back to our original, all right? DNA, we call that as a double helix, and again, all that means is that 2 strands of the DNA are twisted. The 2 parts are, of course, the sides, which is the deoxyribose, the phosphate groups, and the rungs of the ladder, which are the bases. Here we have the phosphate here, the sugar, which looks like that nice shape there, almost like a pentagon. type of shape, and then we have our A and our T that bond together, and our C and our G. And you stack up one of these nucleotides or a bunch of them, and we get our DNA molecule. So what I'd like you to do just for a moment, and this is what you might have to do on the SOL as well, is copy this strand. If this is the one strand of DNA, what would the other side be like? So pause it for a minute. Pause it for about 30 seconds for you to, in your notes, see if you can come up with the other side. So I will start you off. Whoops, I gave you the answers. But I'll start you off. A pairs with T. It pairs with T. G pairs with C. G pairs with C. C pairs with G. A pairs with T. And you just keep on going down the line, making the complementary bases. And this is what you get the other side. Now you see you have a double-stranded. DNA is often called double-stranded because it has two sides of the bases, two sides of the code that match up to one another. So if we move into DNA replication, we end up with two identical DNA molecules that are identical to the original. And so this sounds like mitosis, right? When we go through the cell cycle, actually pre-mitosis, in the S phase, the synthesis phase, chromosomes are copying. And so when we say chromosomes are copying, it means the DNA is copying. And the word replication is just a fancy word to say copy. So if we're talking about most cells of DNA, the eukaryotes, it's found in the nucleus. And it happens before cell division. Okay, so these are all common terms that we have. Again, we're not really talking about mitosis here, it's more the S phase, but you get the hint that it is part of the cell cycle. So it happens in 3 steps. The molecule unzips, we add some base pairs, and we proofread the molecule. 3 easy steps. First step, molecule unzips, just like a zipper. You can see here we have, this is called a replication fork. It looks like a fork, and we use enzymes to do the unzipping. An enzyme called DNA helicase. In many places along the molecule all at once. And then each side we are going to copy. It forms a template, which is just a pattern. So that's the first step. We got to use the enzyme to unzip. Second step, we're going to add the new bases, the base pairs. An enzyme called DNA polymerase is going to help add the complementary bases. A pairs with T, C pairs with G, just like we talked about. So you can see in this picture here as an enzyme is unzipping the DNA. We're going to get one copy on one side, one copy on the other, and we're going to have 2 identical strands when it's all said and done. Last step, though, is to proofread. So if we don't proofread, we end up with mutations. And even so, we still end up with mutations. But anyway, the DNA polymerase, its job is to remove the mismatched bases because nothing is perfect, and some bases pair up with not their correct base, and it's the DNA polymerase's job to catch that. But if it doesn't, so again, back to proofreading. The overall error rate during DNA replication in E. coli is about 1 base in every 1 million, right? So you will get some proofreading errors, and sometimes based on the type of error, and we'll get to that when we get to mutations, that could be significant, it may not. Sometimes errors happen and they're not as significant. Mutations. Before we get to mutations, I want to back up a bit, just show you exactly sort of real time what exactly it is that we're talking about. So I want to scroll down here and go to the steps. So we go here, and again, the first step is DNA replication when the molecule unzips. So here we have it unzips, bzz, unzips, and the second is that we start to fill in these free floaters, and we start to match up the bases on each side to end with 2. So then either step 2 or 3, depending on how you write this, but after the molecule unzips and they pair with a new strand. So here's like fast time. We unzip and the pairs match up, and we end with 2 new strands. One from the original and one where the free floaters, but they're both identical. So when we say DNA is copying or chromosomes are copying, this is exactly what we mean. So now we want to go to the mutation part. And mutations, just by definition, are permanent changes in the DNA code, and that's when copying is being made. the bases don't pair up correctly. And there's different types of mutations, which we will get to at some point. But just know that errors do happen, and this can cause serious effects to an organism. This can cause albinism or sickle cell or any other type of disease. Mutations happen all the time in nature, and that's how things like bacteria, tuberculosis, or any other, even viruses, some can make mutations, become resistant to drugs. So it does play, mutations do play a major role in why we even study DNA. So how is DNA damaged? And when DNA is damaged, copying doesn't happen correctly. So radiation is the most common: UV rays from the sun, X-rays, radioactive elements. Sometimes we have chemicals, pesticides, or cigarette smoke, cause whenever the DNA starts to copy, it might copy wrong. And so we call mutagens anything that causes DNA to mutate, to copy incorrectly. So we'll get more into DNA and the effects of mutations and thing, but these are your notes on DNA and the structure of DNA and how DNA makes a copy of itself. The next step is to take this information and talk about how proteins are made.